Hello everyone, I hope you're doing well. I'm Bhagyashree and this is a course on immunoglobulin structure and function. So let's look into detail. Before that, remember if you like the videos, click on thumbs up button below, and also if you um, stay tuned for more video updates, and if you have any doubts, feel free to ask in the comment section below. Now in our previous lesson, we learned about the different class of immunoglobulin. Just go through that lesson that you will be able to link the concepts. It's over here we are just going to cover a glimpse of it. So different class of immunoglobulin in mammals, there are 5 classes: IgG, A, D, M, E. So we learned in a previous lesson about their functions and about these classes in detail. Then the human subclass for then there are subclass for IgA and G. 4 human IgG subclasses are there, which has got a characteristic feature, except with the variable region. And 90% of homology is there in amino acids for every class, but it is just 60% for IgA and IgG. Now coming to the IgE, it mediates the immediate hypersensitivity reaction, which is responsible for hay fever, asthma, hives, and anaphylactic shocks. And what it does, it binds to the Fc receptor on membrane of blood basophils and tissue mast cells. And then there is a cross linkage of receptor which takes place by antigen, and it induces the degranulation of basophils and mast cells. Now there are a variety of pharmacologically active mediator which give rise to allergic manifestation, and IgE is degranulation which is necessary for antiparasitic defense. Now, today we are going to learn about the action of antibody. So, in addition to the antigen binding antibodies, which take part in range of effector functions and result in removal of antigen and pathogen cells, which is due to the functions from interactions between heavy chain, constant region, and other serum proteins or cell membrane receptor. Not all of the classes of antibodies they have got similar properties. So there is a process called as opsonization. What it is exactly? It is a process by which microorganisms or other foreign particles are coated with antibody or complement and thus they are prepared for recognition and ingestion by phagocytic cells. Now, opsonization of antigen, then IgG bind to Fc receptor, which is on the cell surface which is takes place on the surface of the macrophages and neutrophils. The binding provides phagocytic a way to capture regions, and there is toxin or viral neutralization. So what it is, some positive gram and negative bacteria, they produce extracellular toxins which contribute to pathogenic effects. And there is an immunity against such toxins which depends upon the production of specific antibodies that inactivate the toxins, and this process is called as toxin neutralization. Now once they are neutralized, toxin antibodies complex is either unable to attach the receptor sites on host target cells and is unable to enter the cell, or it is ingested by the macrophages. Now an antibody which is capable of neutralizing a toxin is called as antitoxin. Then the antibodies such as the IgM, G and A, they bind to some of the viruses during their extracellular phase, and what it does, it inactivates them. So this antibody mediated viral activation is called as viral neutralization, and viral neutralization prevents a viral infection which is due to the inability of the virus to bind to and enter its target cells. Okay, so this is it for today. Stay tuned for more lessons to come. Thank you for watching.